Hello everyone and welcome to this webinar. It is a pleasure to see several of you connected to follow this webinar entitled Comprehensive Guide to IMGT, the International Immunogenetics Information System. I will introduce myself. I'm Sofia Kossida, professor at the University of Montpellier and director of IMGT, along with my colleague Véronique Giudicelli, who is a senior staff member within IMGT and the director of the bioinformatics team, will present this webinar to you. I'll switch off my camera so we can better focus on the content of the slides, and I'll bring it back towards the end of this presentation so we can address the different questions. The layout of today's webinar is as follows. We'll start off with some information about upcoming webinars, and then we'll move over to a brief introduction about the adaptive immune response. It will be by no means an exhaustive introduction to the adaptive immune response. We'll focus exclusively to the concepts that will be used when working with IMGT resources. We'll then navigate into the portal, the IMGT portal, before moving over to explore the three main areas of research and development within IMGT. We'll discuss, at the second part of this webinar, we'll discuss the IMGT ontology and the main IMGT standards, and our goal is to conclude these topics within the next 50 minutes or so. At the end of the webinar, we'll address as many questions as possible within the 1 hour time slot. If we exceed this time, all questions asked in the chat will be answered in writing, and the Q&A document will be made available via the very same link that this recording will be made available, and actually it's the very same one that you registered from. The three main areas of research and development within IMGT, as well as the IMGT knowledge graph, will be covered in much greater detail in the subsequent webinars. What is the goal of this series of webinars? IMGT is a rich resource that is constantly being updated. Our goal is to share information and engage further with the community. Therefore, if there is interest in another topic pertaining our activities, do not hesitate to reach out to us to suggest a new webinar. Let's take things from the very beginning, the genesis of IMGT. How was IMGT established? It all started at the 10th Human Genome Mapping Workshop in 1989 in New Haven, during which Marie-Paul Lefranc succeeded in convincing the colleagues that the variable, diversity, joining, and constant genes, which code the antigen receptors, should be officially recognized as genes, as were the conventional genes. Marie-Paul was given the responsibility of managing the immunoglobulin genes. Therefore, she returned to Montpellier in France and established IMGT, which is celebrating its 35th anniversary in 2024. The adaptive immune response is the primary focus of research within IMGT. We specifically study jawed vertebrates or gnathostomata, which range from fish to humans and span 450 million years of evolution. The adaptive immune response is distinguished by its specificity and memory. Here we have a simplified schematic representation that illustrates an overview of the whole adaptive immune response. In essence, B cells express immunoglobulins or antibodies at their surfaces, while peptides of degraded antigens are presented to the T cell receptors via the major histocompatibility complex. This is the so-called trimolecular complex. The three molecule types, the immunoglobulins, the T cell receptors, and the major histocompatibility complex, are central to IMGT's research efforts. At this slide, we have a look at a schematic representation of an immunoglobulin at the left part of the slide. and a schematic representation of a T-cell receptor at the right part of this slide. Each immunoglobulin or antibody consists of 2 heavy chains and 2 light chains. Each heavy chain is composed of a variable domain, we see it here, resulting of different types of genes, namely variable, diversity, and joining, involved in the antigen binding, and a constant domain coded by constant genes, which are responsible of the signal transmission. Similarly, its light chain has a variable domain made up of variable and joining genes, and a constant domain formed by C genes. The heavy and the light chains are held together with disulfide bridges, as we clearly see here. Similarly, the T-cell receptors are made up of variable domains that we see here and constant domains. Theoretically, our body could contain approximately 2 times 10 to the power of 12 different antibodies. This number is ultimately limited by the maximum number of B cells that each one of us is genetically programmed to produce. Nevertheless, it is still a relatively large number, and we'll explore in this slide and the following slides the molecular mechanisms that contribute to this vast diversity. The variable diversity and joining genes that we see here are part of multigenic families that they are organized as clusters at the germline level in most species studied so far. Through a process of gene rearrangement, generally only one gene from each gene type is selected to form the final antibody structure. This mechanism is at the origin of the combinatorial diversity, and this is the first of the 3 mechanisms. Additionally, 2 other unique mechanisms contribute to this diversity, and they are named N-diversity and somatic mutations. We'll explore these 2 mechanisms in more detail in the next slide. We like these schematic representations, and this is once more one of them that we are going to use to describe these 2 mechanisms that we talked about in the previous slide. So we'll describe the rearrangement of the 3 genes at the molecular level and the creation of the junction. This involves the 3' of the variable gene, the complete coding region of the diversity gene, and the 5' of the joining gene. We see these Pac-Man figures that represent molecular enzymes such as RAG complex, Artemis, among others, and they are the ones that they initiate the recombination and trim some nucleotides, as we see here. The exact number and nature of the nucleotides to be trimmed is not predefined. Additionally, there is another enzyme, which is called TDT, which will randomly add nucleotides. This process might seem like a significant energy expenditure. It contributes, however, to the extensive variability observed in the number of immunoglobulins and T-cell receptors. Finally, especially in the immunoglobulins, somatic mutations further increase the overall diversity. So basically, what is the take-home message from this and the previous slides? The message is that there are 3 unique molecular mechanisms that contribute to this great diversity that we are aware of. Having given a brief introduction to the immunogenetics topic, let's now explore what IMGT is all about. IMGT provides detailed data from genome to proteome towards a rich knowledge resource. Here we are having a look at a screenshot of the main IMGT page, and we'll navigate through the main icons at the top of this page. and of course the information provided behind them. We are not going to take them in the very same order as given here. However, we are going to touch upon every single icon. I'll start off with the IMGT universe icon. Behind this icon, information about the IMGT members, the founders, the history of IMGT, as well as information about our dedicated scientific advisory board that officially started at the end of 2023, while in the spring of 2024 we had our first on-site meeting, can be found. We move over to the IMGT knowledge icon. Several pages with relevant to the field content are provided behind this icon. For instance, the IMGT scientific charts that mainly provides the rules used by IMGT ontology, as well as the IMGT standards that we'll hear much more about it during the second part of the talk by Véronique. We see the IMGT index that provides an alphabetical list of the most important terms along with their definitions used within IMGT. The biotechnology as well as medical and veterinary pages provide relevant information for the antibody engineering field, whereas the IMGT references is the section that includes the whole list of IMGT scientific publications as well as how to cite the different IMGT resources. Creations and updates icon. Here we find meticulously maintained pages that provide the necessary information for traceability within IMGT databases, as well as news at which users can subscribe. IMGT offers an RSS feed to which you can subscribe via this URL and use one of the many freely available tools, one example is provided here, to manage the news. The databases icon, I suppose it's a self-explanatory icon that provides information and lists the databases that they are available within IMGT. In this slide, I'm providing the databases that they are accessible via the interface, interconnected. They can be queried according to IMGT ontology and are downloadable. So basically, I'm giving two words about IMGT LIGMDB database, which includes sequences from different steps of immunoglobulin and T-cell receptor synthesis. And therefore, this database, which is a nucleotide database, integrates three main categories of sequences. The first one is large germline non-rearranged genomic DNA sequences, which may include a complete locus for several hundred kilobases to one or more megabases. The second category includes rearranged genomic DNA sequences resulting from the recombination of VJ genes or VD and J genes. And the third category includes rearranged complementary DNA sequences. Most of the IMGT LIGMDB nucleotide sequences come either from GenBank or from ENA, using the very same accession numbers in order to facilitate interoperability with the generalist nucleotide databases. The curated immunoglobulin and T-cell receptor genes that are entered and managed in IMGT GeneDB with all IMGT identified alleles highlight the potential great polymorphism of these genes and can be retrieved via this database. The tools icon. It's another self-explanatory icon, I suppose, where the tools are available within IMGT, that they are accessible via the interface and help users analyze their data by comparing them with the reference directory. This icon provides these tools. So I'm going to touch upon really briefly on vQuest and IVQuest, that they are the tools for the analysis of the nucleotide sequences via the interface. I'm going to talk about IMGT domain gap align, which aligns the amino acid sequences per domain, creates gaps according to IMGT unique numbering, and highlights differences with the closest reference gene cell alleles found in the IMGT reference directory. All of these tools, they will be addressed in much more detail in the subsequent webinars, whether the different axes of research and development within IMGT will be explored. The IMGT repertoire icon. The information hidden behind this icon is quite rich, and information is provided for the immunoglobulins and T cell receptors, and it will be explored in much more detail in the following webinar, which is the webinar dedicated to the biocuration. Several categories are provided, such as locus and genes, proteins and alleles, 2D and 3D structures. The final icon, for which I have not created a specific slide, is the downloads, and basically is the place where from the data in different formats can be downloaded. Having concluded the little description of the icons at the top of the main IMGT webpage, I'll move over to the second part of my talk, which will be to provide an overview of the research and development activities within IMGT. There are three main areas of research and development, and we are going to spend a couple of minutes to describe and explore a bit more each one of them. Deciphering the immunoglobulins and T cell receptor genes in the genomic loci of jawed vertebrates is the first axis of research and development within IMGT. So basically we're going to have a look at this poster, at the first part of the poster, which is the jawed vertebrates, as we already mentioned, going from fish to human. We take as an example the human species, and the human DNA assembled into chromosomes is accessible through publicly available databases. The 7 immunoglobulin and T cell receptor loci are located on 4 chromosomes, namely chromosome 2, 7, 14, and 22. If we have a close look at the telomere of chromosome 14, we see a black stripe that corresponds to the immunoglobulin heavy locus. We see here a graphical representation of the IGH locus, which is composed of the multigenic families, as we already said, of the variable, diversity, and joining genes. The IMGT biocuration pipeline, which is detailed in scientific publications and is assisted by the IMGT LIGM motif software tool, which is an internal tool. It's an internal tool, however, there is a version that is available online as well for users to use, and there will be soon an updated version of it available. So basically this tool is designed to identify and precisely characterize these genes. It's a semi-automated procedure, the biocuration pipeline. The curated data of genes and alleles are available through the IMGT GeneDB, as mentioned previously, while the locus nucleotide sequences are added in IMGT LIGMDB, an IMGT reference directories, basically, because there are several of them, are provided to the scientific community and integrated in IMGT tools dedicated to the analysis of the reference sequences. We are going to have a look at an ongoing IMGT biocuration project, which is the IGH analysis across diverse human populations. So basically, we are going to explore this schematic representation of this slide, and on the left slide, on the left part of the slide, we have a look at the human IGH locus, which is depicted here with 7 identified copy number variations. We see them here: 1, 2, 3, 4, 5, 6, 7 CNV forms. In the center of the slide, we zoom in on the third CNV form, and what basically we are having a look at is that there are several individuals from different geographic representations that they represent different human populations, and we have a look at the different forms of the CNVs. Whereas on the right part of this slide, we see a schematic representation of the haplotype, and we define the haplotype. The haplotype refers to the unique combination of the CNV forms present in each individual and the upcoming database and the upcoming haplotype database. In this slide, we present a non-exhaustive, recently completed or ongoing IMGT biocuration projects. So basically, many of these projects, which results from successful collaborations, are listed here. And apart from the human assemblies that we saw at the previous slide, the IMGT biocuration efforts have also included several non-human primates. The gorilla project that was accepted for publication 2 days ago, the rhesus monkey, lemur, the orangutans, as well as some other notable projects like the mouse inbred strains, horse, necked mole rat, which was recently published, the ferret, which is submitted, and the different dog races, among others, obviously enough. And I will go ahead and conclude the first axis of R&D within IMGT by providing some recent developments for and with the community within this axis. So basically, data, IMGT data, they're accessible via NCBI, TPA, third-party annotation, and recently there is a collaboration with VGNC for the annotation of the Labrador dog, the IG Kappa, that will be available via VGNC. We are publishing the germline reference sets, and the nomenclature rules are available. All the details of them are available via our IMGT web pages. If you go to the news, they were recently released. The URL is available there. Once more, I'll underline the fact that we are open to collaborations. Of course, we are limited by human resources, but don't hesitate to reach out to us to prioritize the different biocuration projects. We now turn to the second axis of R&D within IMGT, which focuses on exploring the expressed, this time, immunoglobulin and T-cell receptor repertoires. So basically, we are having a look at this poster once more. This is a schematic representation. As always, we are discussing the adaptive immune response. And this simplified upper part, we address rearranged DNA or RNA sequencing of Ig and TR. And this is most of the times work that is carried out before we come over to IMGT. IMGT provides software solutions, namely IMGT VQuest and its high throughput version, IMGT High VQuest, in order to analyze these expressed sequences using the reference directories that we described in the first axis. Fundamental questions in basic research on the adaptive immune response, clinical research queries, as well as antibody engineering are among the themes that can be addressed using the IMGT HiVQuest tool. There will be a dedicated webinar to address the nitty-gritty details of these tools. However, here we have a look at the interface of IMGT VQuest for the analysis of up to 50 sequences, which uses the same algorithm as the IMGT HiVQuest, and therefore users are encouraged to test their sequences using IMGT VQuest before transitioning to IMGT HiVQuest. At this slide, we are having an overview of the main functionalities of IMGT VQuest, which include identifying the germline variable diversity and joining genes and alleles, delimiting the framework and the CDR regions, analyzing the junctions, describing mutations both at the nucleotide and amino acid levels, and providing a comprehensive annotation of the V domain. This slide presents the interface of IMGT HiVQuest, which is, as we said, the high throughput version of VQuest. Therefore, up to 1 million sequences can be analyzed. This interface is the only interface that requests user registration. Results can be, the output of the results can be given in CSV formats as well. I'm not going to go into the details of these tools as they will be the topic of dedicated subsequent webinars. This slide will conclude the overview of the second area of research and development within IMGT. We are now moving to the third and final axis of R&D within IMGT, transitioning from nucleotides to amino acids. So basically, if we focus at this upper part of the poster, we see receptors, chains, domains, amino acid sequences, and contact details that are analyzed, and the data are integrated within the IMGT 2D structure DB, IMGT 3D structure DB. IMGT Collier de Perle is a tool for 2D visualization of immunoglobulins and T-cell receptor proteins, while the IMGT domain gap-align tool, as we already mentioned, serves comparing a query with the IMGT reference directory and performing domain localization, as well as looking in detail in the amino acid changes. In this slide, we are going to explore the main functionalities of the databases that we mentioned, and we are going to explore the main sources of input data, which are basically two. There is the PDB, the Protein Data Bank, and the International Non-Proprietary Names List, which is issued twice per year by the World Health Organization. As we said, the bi-curated data from IMGT are incorporated into IMGT 2D and 3D structured database, and they are also linked to the monoclonal database IMGT MapDB. I will conclude the overview of this third axis of research and development within IMGT by giving an overview of some research projects. So basically, the data processing, there is based on machine learning methodologies. We have automated the feeding of IMGT MapDB. There is a standardized way of presenting the mechanism of action of the monoclonal antibodies within IMGT that is available that is available via the database, and it can also be queried. There is an article describing the mechanisms that they have been described from publications so far. And the purpose of this one is much more for pedagogical reasons. The project on the FC region, which is the FC region that is responsible for effector functions and exchanging amino acids at key positions to manipulate the interactions and modulating cytotoxic effector functions are currently major areas of focus in several labs. So basically we'll soon provide a tool that can list these mutations. And I'm gonna conclude by touching upon a tool that is available via our web pages that helps the prediction of binding and interaction both of class 1 and class 2 major histocompatibility complex and peptides resulting from antigen processing. I encourage you to check out the tool. There will be a dedicated webinar for this one as well. So basically this concludes the first part of this webinar. I'll leave the floor to Véronique Giudicelli, who is going to address the second part of this webinar. Thank you very much.
Thank you, Sofia. I'm Véronique Giudicelli, and we are moving to the second part of this webinar. Since its creation, IMGT has developed standards to enable researchers and clinicians to speak the same language, and in order to maintain this consistency across the different components of IMGT, which cover a large spectrum of data types from genes to proteins and antibody engineering. I will review the basic concept of IMGT ontology, the first ontology in the field of immunogenetics, and the IMGT standards. We will see examples of identification concepts with standardized keywords used in IMGT databases and tools, and in particular with the terms dedicated to the evaluation of this functionality. The classification concepts are fundamental for the setting of the IMGT gene nomenclature. The description concept with the IMGT labels allow to describe any motif of biological interest in sequences and structures. The numerotation concepts are the rules for the IMGT unique numbering for variable and constant domains of immunoglobulin and T-cell receptors. The localization concepts correspond to terms that allow to localize a set of genes in the genome, and the orientation concepts are terms for the orientation of entities on chromosome, loci, and sequences. You will find in the slides the corresponding links to the IMGT web resources which detail these concepts and standards. We are starting with the concept of identification. On this slide are shown the different steps of the immunoglobulin synthesis in humans. We have defined molecular entities which identify genes and rare sequences at any step of this synthesis, and this in relation with the type of molecule in which they can be identified, gDNA, mRNA or cDNA, or proteins, and in relation with the type of configuration, which is germline before any recombination of V, D, J, or V and J genes, or rearranged in otherwise. For example, a VDJ gene corresponds to the rearranged sequence of 3 types of genes, variable diversity and joining, that can be identified in genomic DNA during the synthesis of an immunoglobulin-heavy gene. The molecular entities can be used to request the databases of IMGT and to extract corresponding sequence, and more generally the exhaustive list of the molecular entities with the exhaustive list of all other keywords of IMGT that can be used to request the database are available in the IMGT scientific chart page on the IMGT website. A second example of identification concepts corresponds to the terms that determine the functionality of genes and of reference sequences. Three terms identify the functionality of genes. Functional if the gene comprises all constitutive motifs as an open reading frame and a coding region without any stop codon. Pseudogenes show major defects, and the functionality is identified as open reading frame, ORF, for genes with small defect in their germline sequences and for which we do not have any information on the expression of the gene as protein. For information, here are the color code for variable diversity joining and constant genes according to their functionality that you will find in the IMGT locus maps on the website. In the case of reference sequence, two terms were needed. Productive identifies the functionality of a reference sequence with an open reading frame, without any stop codon in the coding region, and with an in-frame junction between the V and the G genes. Otherwise, in case of stop codon, defects, or an out-frame junction, the sequence is identified as unproductive. The detailed rules for the determination of the gene or sequence functionality are also provided in the section 1 of the IMGT scientific chart page on the website. We will illustrate the concept of classification with the scientific criteria used to classify immunoglobulin and T-cell receptor variable genes. Here is shown the map of the human immunoglobulin lambda locus. All rectangles in red, green, and yellow correspond to variable genes colored according to their functionality, as we saw in the previous slide. These genes show similarities and are also highly polymorphic. Let's see the criteria used to classify these genes and to define their names according to the rules of the IMGT nomenclature. IG and TR genes can be classified into groups, whatever the species. A group brings together the genes that code for the same chain type, here IGL, and that share the same gene type, here V for variable. In a second step, we are defining the concept of subgroups. Subgroups of variable genes. For a same species, the variable genes which belong to the same subgroup show more than 75% of identity in their coding region, the V region. Then we have the concept of gene, which is characterized by its position within the locus or in the V cluster. We have the example of the gene IGLV211, which belongs to the subgroup 2 and is in 11th position within the locus and among the variable genes starting from the 3' end of the locus. The first allelic form of the gene is named *01. Then, if new alleles are identified, same gene, same position in the locus, but with variation in the V region, the number is increased, *02, etc. The classification concepts are defined in the index of IMGT, and the rules for the IMGT gene nomenclature of any immunoglobulin and T-cell receptor genes are detailed in a dedicated section of IMGT scientific chart page. The third set of concepts, the concepts of description, comprises the IMGT labels written in capital letters for the description of the constitutive motifs of immunoglobulin and T-cell receptor sequences and structures. In addition, IMGT has defined a standardized graphical representation of the sequence for all molecular entities presented before, whether as a gene, transcript, or protein level, and these representations are called prototypes. The prototypes show all IMGT labels that should be described in a given entity. Here are shown the prototypes of two entities described in genomic DNA, a variable gene in germline configuration, and an entity resulting from the arrangement of V, D, and J genes. We can see the different labels and the common labels, especially regarding the variable gene. All the IMGT labels are used for the annotation of nucleotide and protein sequences by biocurators and by IMGT tools, and these labels can be used to request the databases. Thanks to these representations, it is clear that these labels are linked together with specific relations that organize them in space in a given prototype. Actually, the several relations illustrated in this slide are necessary and sufficient to describe all types of relations between the labels. For example, L part 2 and FR1 are contiguous, and they are linked by the relation A meets B. Here are shown the identifiers of the concepts of relation ontology that correspond to equivalent relation in IMGT. Indeed, we introduce mapping with other external ontology to highlight these correspondences. The list of IMGT labels with their definitions are available from the IMGT scientific chart page on the website. The concept of numerotation provides the rules for the IMGT unique numbering of variable and constant domains of immunoglobulins and T-cell receptors, whatever the species, the receptor type, or the chain type. Let us take the example of a variable domain. Here is shown in these slides a 2D graphical representation of a variable domain of an immunoglobulin heavy chain. This graphical representation is known as IMGT collier de Pearl. According to the IMGT unique numbering, the amino acids or codons that are conserved, whatever the species in a variable domain, are always at the same position. For example, the cysteine 23, the tryptophan 41, the cysteine 104 of the V gene, and the tryptophan 118 of the J gene. This IMGT numbering allows the standardized delimitation of the framework F4 represented in black beads FR1, FR2, FR3, and FR4, and of the complementary determining region CDR, CDR1, CDR2, and CDR3. IMGT gaps are shown by hatched circles in IMGT collier de pearls. They correspond to the unoccupied position according to the IMGT unique numbering, and in particular, this allows to take into account and to represent the different lengths of the CDR1 and CDR2 that can be identified in germline genes at the DNA level. On the website, we have a dedicated section to the IMGT unique numbering, which can be found in the IMGT scientific chart page. In particular, IMGT provides in this section tables which indicates the missing position according to the length of the CDR1 and the CDR2. For example, for a CDR1 of 8 amino acids, the positions 31 to 34 are not occupied. The unique numbering of the CDR3 represents a different challenge. Indeed, it results from the arrangement of 2 or 3 genes and shows a high variability in its amino acid length. In the IMGT unique numbering, the default length for the CDR3 is set to 13. However, for shorter CDR3, gaps are introduced on the top of the loop in sequences and in collier de pearls, and for longer CDR3, additional positions, as here for 17 amino acid lengths, are introduced at the top of the loop. By convention, the CDR lengths of CDR1, CDR2, and CDR3 are shown between square brackets separated by dots, and it is important to note that the IMGT unique numbering is a crucial concept of IMGT, as it represents the reference for the description of the allelic polymorphism in immunoglobulin and T-cell receptor genes, for the description of somatic mutations in rare sequence of immunoglobulins, and for the comparison of variable and or constant domains across different species. The concepts of localization allow to characterize the specific localization of sets of immunoglobulin and T-cell receptor genes in the genome. The main concepts of localization are the locus, which is a set of Ig or TR genes that are ordered and localized in the same chromosomal location for a given species. A locus comprises genes from different groups that could be expressed in the same gene type. A locus is characterized by its length, its cytogenetic chromosomal localization, its orientation, and its gene content. A chromosomal orphan set is a set of Ig or TR orphans, which are genes outside the main loci, localized on the same chromosome in the same location for a given species. Chromosomal orphan sets comprise genes which are not expected to be expressed as protein. Within a locus or chromosomal orphan set, the gene order is the relative position of a gene, whatever its gene type, from 5' to 3' of the locus. All known genes in a locus are ordered according to the gene order. Even the non-Ig and non-TR genes present in the locus, because their position may be useful for the gene mapping. The gene order allows to highlight the polymorphism by insertion and deletion in the locus and the copy variation numbers, and it allows to highlight also unusual features in a locus in a particular assemblies. The concepts of localization are described in the IMGT index. The concepts of orientation at the genomic level specify the orientation of an entity on a chromosome and in a locus. The orientation of a chromosome is defined as forward from the short arm to the long arm. Therefore, the orientation of a locus or a gene on a chromosome is defined as forward if the orientation of the transcription is identical to the forward orientation of the chromosome, or reverse otherwise. In the two examples, the human IGL locus on chromosome 22 is in the forward orientation, whereas the human IGH locus on chromosome 14 is in the reverse orientation. Regarding the orientation of a gene in a locus, regardless to the chromosome orientation, two terms were created. The orientation is said direct if the entity is in the orientation of the locus, or opposite otherwise. The example here, IGK-V41, is in the opposite orientation in the IGK locus. Besides the genomic orientation, the DNA strand orientation concepts describe the orientation of genes, motifs, labels in sequences. Sequences are by default orientated in the sense 5' to 3'. Entities are said sense or plus if they have the same orientation as the sequence, or antisense or minus otherwise. Please note that in the case of antisense entities, the sequence has to be complementary reverse to get the correct coding region. The concept of orientation are detailed in the IMGT index on the website. We would like to add to this short introduction of IMGT standards the presentation of the 11 classes of amino acids defined by IMGT. They are based on the three major properties: hydropathy, volume, and physicochemical characteristics of the amino acids. These classes are particularly useful to qualify any amino acid change in the case, for example, of non-silent hypersomatic mutation in immunoglobulin sequences or in the case of artificial changes in the context of antibody engineering. Conventions have been set, for example, from a change from valine to leucine. The hydropathy has been preserved, plus the volume differ, minus, and both amino acids have the same physicochemical properties, plus. Because this change maintains two properties, it is qualified as similar. The evaluation of the amino acid change is implemented in IMGT tools dedicated to the nucleotide and the amino acid sequence analysis, and the details of the amino acid classes are available in the aide-mémoire of the IMGT website. To conclude this part, we had an overview of the most important IMGT concepts and standards, which are implemented during the annotation processes, as well as in the IMGT tools. You will discover them in more detail in the presentation of the next webinars. Taking advantage of the new technology associated with the deep learning and the semantic web, IMGT has developed a knowledge graph, IMGT KB, shown in this slide, which integrates IMGT ontology and external resources. This integration highlights the relationships between data from the different components of IMGT, highlights the mapping with external ontologies, and allows to explore potential new relationships and extract new knowledge. IMGT KB will be presented in a dedicated webinar. Well, this first IMGT webinar is almost over. We thank you again for your presence and your attention, and we will be happy to answer your questions.